Good afternoon, and thanks everyone for joining our panel. I'm Geulah Livshits, a biotech analyst at Chardan. So the AAV gene therapy space continues to mature, both in terms of programs transitioning to late-stage studies and in terms of advances in manufacturing, process, and analytics. In this panel, we'll explore the evolving state and standards of AAV gene therapy manufacturing, what regulators are looking for, and what learnings can be gleaned from prior experience in the space. So today with us on the panel, we have Zandy Forbes, President and CEO of MeiraGTx, Frederick Porter, CTO of Taysha Gene Therapy, and Pierre Caloz, CEO of uniQure. Thank you, all of you, for participating. So the format is a roughly 45-minute panel discussion. If any of our audience attendees have questions during the session, feel free to type them into the question box underneath the video player, and we'll try to ask them on your behalf. So to start off, will the panelists please each take a moment to introduce themselves, their company, and their company's manufacturing capabilities before we discuss the space in more detail. We can start with Zandy.
Thank you, Geulah. I'm the CEO of Meira Gene Therapies, and we've really focused on manufacturing since we started the company in 2015. So we have multiple GMP-approved manufacturing facilities, GMP-approved facility in London, which is flexible and scalable, an additional facility in Shannon Island, which has viral vector suites onboarding this year and has an up and running plasmid facility to supply GMP, plasmid for GMP. We have a process development facility where we not only have developed a flexible process for manufacturing AAV, but we also develop assays, and we are bringing in-house all, I would say all by the end of this year into next year, of the assays required for QC and stability and release of our products manufactured at our facilities, as well as fill and finish, obviously.
Fred, you can go ahead.
Okay. Good afternoon, everyone. I'm Fred Porter. I'm the Chief Technical Officer at Taysha Gene Therapies. So Taysha is a young gene therapy company focused on diseases of the central nervous system. I've been in the gene therapy manufacturing field since the late 90s, started out with adenoviral vectors, worked in viral vaccines of multiple different viral platforms, and now at Taysha, we're advancing adeno-associated virus gene therapy candidates to clinic. Our approach at Taysha as a young company really is a multi-prong approach. As a company, we are a direct spin out of University of Texas Southwestern Medical Center, and we partnered with them very early on in some of our early manufacturing efforts. Since then, we've then gone on to partner with more established contract manufacturing companies in the space, namely Catalent and ViralGen, to advance some of our early stage programs, but also at the same time invested in establishing our own commercial manufacturing facilities and process development and analytical development laboratories, all in North Carolina. And I'm happy to talk about our story and our thoughts on gene therapy manufacturing during the discussion today.
Pierre?
Thanks, yes. Good afternoon. My name is Pierre Callow. I'm the Chief Operating Officer in uniQure. I'm actually based in Europe, but in uniQure we have two sites, one in Amsterdam, where we are headquartered. At some point we are a spin-off of the hospital from Amsterdam, and we have also a facility in Lexington near Boston. We have about half of our people in Europe, mostly on the research and development side, and we have a commercial manufacturing facility in Lexington with about 220 people, a bit more on every site. I'm actually responsible for operation, which means for us process development, manufacturing, supply chain, engineering, as well as CMO management, as well as CMC portfolio development. So this is quite a big group. We are multi-product. So uniQure, maybe you're aware of uniQure. uniQure is mostly insect cells based, so baculo system. And we had our first product approved in Europe, which was Glybera, which was the first product approved in Europe that we don't produce anymore. And we have several programs, including hemophilia B, that we are partnering with CSL at the moment, so CSL Behring. And I'm also happy to be here and discuss about manufacturing.
Perfect. Glad everyone's happy and excited to discuss manufacturing. So with intros out of the way, let's kick things off by getting right to it. I'd like to get your thoughts on what is involved in taking an asset from academia and then translating it to the commercial phase. So let's start with discussing any differences that you've noticed in manufacturing standards for early versus late stage programs. And this time, let's start with Fred, then go to Pierre, and then Zandy.
Yeah, I think this is a really relevant topic to almost every company in the field. Most of the innovations in identifying new targets really come out of an academic partner's laboratory. And I think the way we like to think about it at Taysha is really beginning with the candidate itself. So I think that's really important, is not only to think about manufacturing, about the candidate and what information we have about that candidate that's really been established in an academic laboratory. So understanding reagents, and know-how that's really been established using academic manufacturing processes, and really trying to pull that forward into our manufacturing strategy. So what we've tried to do at Taysha is, move as quickly as we could, as we can, to our what we expect our clinical phase manufacturing process will be, especially for pivotal stage efficacy and safety materials. And so that's where we've really invested heavily, understanding the construct, making sure the construct is well-defined as it moves into manufacturing, and then using that process, that platform process early on, that we could envision using commercially in the end for commercializing this product. And that's what we've tried to do at Taysha. We've established a suspension-based HEK process that we are using across the board for all of our candidates, and we're using that process for those early clinical candidates and materials as quickly as we can. Another thing to note is really using analytics very early on that we think can help us characterize those products to make sure we have a good product profile, and that really is the basis that we discuss our product with the agency. And finally, I'll just, to give other panelists an opportunity to speak about this as well, is retaining critical early materials. So any viral vector that was used for some of the early developmental studies, retaining that in case we have to do additional comparability or analytical work to understand differences in manufacturing going forward. So that's kind of what we've been doing at Taysha. We've been advancing multiple candidates from academics into clinic over the last 18 months. And so that's kind of the basis of our strategy.
Great. And Pierre?
Yeah, there's some convergence here, I guess, with what Fred was saying. Indeed, this is really key to be able to start with the end in mind. So at uniQure, we have product that we are bringing to the market, and we have some very early programs. And the name, the term platform was already mentioned once. Everybody's talking about platform. This is one of the key aspects. Everybody's talking about platform. I'm not sure we all mean the same. We've put quite a lot of energy to define what our platform is. So we have broken down our platform into modules. We have people that are in charge of those different modules, and we make sure that we know what our platform is, what is the basis for development, what is the platform that we can capitalize on, but how we can also innovate based on the platform. So for one early program, we may use the platform generation 1 or 2, which is not fully optimized, in order to generate rapidly material, right? This is about speed, this is about proof of concept, this is about, but still we need to be able to have something which will be representative for later stage. And later on, you need the platform to be robust, compliant, have the right cost of goods, right? This will be, and to give the same product at large scale. So part of the secret is probably about the platform. And indeed, one of the modules we have in our platform is the analytical toolbox. It is really important that from the beginning, you start to have the right glasses on, such as you can characterize what you do at small scale, mid scale, or large scale. Having said that, we tend to use a large scale as early as possible, or to use directly the platform at the scale that we'll use later, very early on. So just a couple of inputs.
Great. And Zandy?
Yes, so we have early on taken some academic programs through the clinic, which are in phase 2, and currently we have a pivotal program that originally came from academia. Very early in setting up the company, immediately we did build facilities. We've spent probably more on any other aspect of R&D in our process development, developing a platform process. And what I mean by that is that at the 3 unit operations, from upstream all the way down to the separation and final product, we optimize each of those with multiple products, multiple capsids, multiple viral vectors. So we have a process which in our facility is continually being optimized, but can be rapidly applied to every single viral vector that comes through our pipeline. Now, as everyone's mentioned, is academic manufacturing tends to be very different from what might even be acceptable to an IND. So our platform process, which we use for our pivotal and potentially commercial product, is the very same that we now use for all of our INDs. And we fit that to any viral vectors we get from academia. But on the whole, most of our new products are actually optimized subsequent to seeing data from academia. So we have a vectorology platform that looks at every single aspect of those viral vectors, in the promoter, the sequence, everything, to optimize it for potency. And at the same time that we're doing that optimization, we're also looking at manufacturability. So we don't consider one to come after the other. But as we're making the very sequence of our vectors, we're looking at how they behave in our process, what modifications to the process to give best yield and quality, and how to actually turn it into a product. If you have different choices, one which is very easy to make and one that isn't, we look at all of those factors when we're choosing the final viral vector that we're going to take forward into the clinic.
Mm-hmm. Got it. And so in thinking about the challenges for manufacturing at different stages, to what extent are there different bottlenecks for commercial manufacturing versus early? And can you discuss some of those? And let's start with Pierre, and then move to Zandy and then Fred.
Yeah, I mean, the obvious one at the moment is probably supply chain. Even for basic things, supply chain is an issue in the world today. So certainly in the gene therapy business, this is the case. I must say this is quite frustrating at the moment to have a process which is, highly technologically developed. Can you say that? Great science, great products, and you are stuck because you cannot get a piece of bag or a piece of plastic tubing. So supply chain is really an issue. And actually, we need to think about that in development, because we may have used in the past some customized assembly, for instance, and we need to move towards more ready-to-use systems, such as we are not bound to a single supplier. So supply chain is certainly one of the big bottlenecks today. I think we mentioned already the analytical toolbox. The analytics is a big a big issue, is a big bottleneck. So that's a bottleneck in terms of, it is very costly and it takes a lot of time to develop the right analytics. But also sometimes those testing are very long to do. So you need to think about what are the release testing that you use in routine, what are your extended characterization analytics. Otherwise, if everything is within the same bucket, then it is a big bottleneck once you need to produce one run per week. I would say in general, we tend to have in gene therapy a lot of scientists, PhD, a lot of experts, but we lack in terms of talent, biostatisticians, supply chain experts, engineers, and not only experts, but people who understand a bit of, not only one discipline, but more than one discipline. So you need good regulatory people who understand engineering, you need engineering who understand science. It is the same also for large molecule, for monoclonal antibody probably, but this is even more acute in the gene therapy business. So this is three main bottleneck we are experiencing today, with a very competitive market, certainly for talent. So yeah, just a few points.
Great. Zandy?
Yes, so I think we can look at supply chain in a big way, and also a less macro way. And clearly it can be extremely frustrating, particularly at the moment, not to get equipment. I will say that our manufacturing team has excellent supply chain management, so that over the last 2 years, we have been able to produce product for 5 different clinical programs in our facilities. At some of those times, our supply chain guys were helped by Janssen, our partner, who were very helpful. But it really, to your point of finding people who are good in supply chain, that's one of the areas of manufacturing, our CMC group, that I'm most proud of over the last 2 years, because it's a huge job and it really takes a huge amount of focus to make sure that you have 12 months of everything in advance, which is what we try to do for all of our, and there are thousands of these components. It's not just 5. So it's a really important and really big job. From a more macro perspective, the capacity for plasmid production, for triple plasmid transduction, that is quite limited. And we were starting not only to see big costs, but lack of flexibility and timing issues with getting good quality plasmid for GMP. So we did build our own plasmid facility that now currently is probably scalable, such that it can scale to probably 10-fold more than we and our partners use it in our current pipeline. And then over the last couple of years, as we've been preparing with our partner Janssen for a potential BLA of our pivotal program, it's become very clear that the analytics, the QC for stability and release are incredibly limiting. So at our facility in Shannon, we're bringing in full QC for release of all our products. And we obviously initially were doing potency assays and all of those different analytics, but it's just very, very difficult out there to not only get the timing, but the quality. You've got to have a reliable assay in order to show your material is reliable. And I think we're at a moment where we're having to bring that in-house to a large extent to get that reliability of time, of quality, and of cost. So we've gone to both ends of the supply of the product and brought it into Meira.
Great. Fred?
Yeah, thanks. Totally agree with the comments that Zandy and Pierre made, but I thought maybe I'd take it in a different direction in thinking about, what are the bottlenecks getting from an early clinical candidate through to commercialization? It's really, striking that balance on the continuum. As manufacturer of our clinical candidate, our teams are asked to move quickly to get that first in human batch, right? So it's setting that process that is good enough, that is representative of where we'd like to go commercially, early enough that we feel like we're not making too many changes between then and that commercial pivotal batch. And I think the way that we're thinking at Taysha about early and late stage, in traditional drug development, phase 1, phase 2, phase 3, everything's smashed together here. We're really thinking early clinical manufacturing and late in PPQ. And so it's trying to get that right, that balance of speed and representativeness, not around just the process, but the methods. And some of the real methods of note are potency assays. All agencies are expecting a product-specific potency assay. We know that many of the gene candidates that were asked to advance to the clinic might not be enzymes, might not have an easy to assay functional activity. And so what we're doing at Taysha is investing really early on in what does that mechanism of action assay look like, because that often can become rate limiting for building that panel. Regulators are expecting a potency assay to release that pivotal stage material, and it can take years, right, if you don't invest early enough. And so that's one thing we're doing in parallel with the track of manufacturing product, optimizing the process, is really investing in that and having our analytics in the appropriate stage of qualification or validation at that pivotal stage, and to be able to have those really meaningful product characterization conversations. So totally agree with what the other panelists mentioned, but other things that we're thinking about too. And I think we're fortunate at Taysha that all of our candidates are AAV9 capsid based. So we do have an assay platform that we're really refining with program 1 that we apply to program 2 and really getting it to a really good stage of development. I will also give a plug for characterization assays. We're focused on the release panel. We all know we must do that. But what we've found in our conversation with the agencies, it's the additional characterization that wins the day. And so investing time and money in that is something that we found is really important.
Great. All of those really interesting points. And so kind of building off of what Fred was just talking about, advancing from early to late stage is often involved incorporating changes to manufacturing, and whether that's going from adherent to suspension, some other bioreactor scale up, or other kinds of process optimization. So from your perspective, when is the right time or the best time to incorporate changes to transition from a commercial process? And in particular, what do regulators want to see in terms of bridging or comparability studies, and what are some learnings that can be taken forth from your experience? And let's start with Zandy, and then go to Fred, and then Pierre.
Yeah, so I think, the real truth is, in an ideal world, you'd file your IND with your commercial process, right? But as we've mentioned, there are balances, of a number of things that may not make that happen. I would say over time, as you iteratively optimize your platform for multiple viral vectors, we get closer and closer to actually at IND having something which is pretty close to commercial, which is great. In the same facility, the same cells, more or less the same process. So, and very importantly, you always retain material, and the analytics remain the same, so that you can always show comparability from your earliest lots and what was originally used in tox, all the way through potentially, iteratively to your approved product. That's the ideal. I would say that the most important thing is to be able to show the agency as you improve, because you're not always wanting the identical product. You may want to improve it over what happened in the phase 1, potentially. So really the very important thing is to be able to show that you've changed the process with the aim of improving feature A, and you have made it reproducibly better or equivalent to the original. So very important to constantly be using the same validated assays from the earliest time points all the way through to the end to constantly show and reassure the agency that you're in control of your manufacturing and you're manufacturing what you say you're manufacturing at every single step. And you can show the link between the first and the last.
Right.
Yep. I can't agree more. I think we're always stuck in a place of compromise. Trying to have that final process as early as possible. But I think there are a number of reasons why that's not always the case. You might inherit a program. You start with something that is not your standard process. So in terms of comparability, though, I can't agree more. It's really looking at the product quality attributes feature by feature. And I think we've found that agencies are receptive to have those conversations if you can put together a proposal really focusing on those quality attributes of the product. And those are the biochemical and molecular features of what we're making in gene therapy, which is quite complex even among biologics. I mean, it's a bit of a mashup of a protein biologic and a molecular nucleic acid biologic. So you need analytics that come at it from both ends, where in a protein field, it's mostly protein analytical panel or a molecular panel for, let's say, something that's plasmid DNA, messenger RNA. We have to do both. And we need to have not only the most robust, reliable release assays, but those characterization assays. We found that showing control, multiple replicates, if we want to argue that we have a comparable product or process, being able to show multiple lots with the new process, whether they be GMP lots, engineering lots, or development lots that are representative, is a really helpful strategy to show that you have a process in control, that it's performed across scale, I think is really helpful. And it's really focusing on those quality attributes. Obviously, things like pH we must measure. Is that really part of the comparability argument for a product? Yes, in terms of process performance, but we're talking about the product itself, and those are the assays we're really honed in on. And potency assays and potency and activity are really central to all of that. That's what our thought is and our approach.
Yeah, this is good to see that we all agree. Frankly, at some point we don't want to make change, right? This is, if we can avoid change, that's a big deal. So therefore, this platform approach is key. But indeed, it does help to have all those acronyms like the QTPP and the CQA and the CPP and this approach of quality by design, where you can link the target product profile to your critical quality attributes, to your parameters. I mean, if you can show that you understand this link, this helps a lot. However, even when you define that very nicely, when you have the right analytics, this is not always easy to explain small differences. This is not always easy to explain what is better or not better, right? This question of empty, full, for instance, some people would argue that some empty is actually good, and some people would say that empties are actually something that you should get rid of. So this is still early days. So again, if we can start with the right platform very early on, that this is better, which means that the manufacturing guys need to frustrate a little bit the research people. I think we need to come up with some kind of menu and say, this is what we can do. These are the options that we have in our platform. And we need somehow to stick within this list of options. We cannot just do everything and keep complete agility until the end. This is something which is not doable, even if we would say, yeah, but then let's show that the CPP has no impact on the CQA, and we'll have, I mean, this becomes quite difficult. So at some point, you need to restrict your level of creativity as you develop your process for later stage.
Got it. That makes sense. And so thinking about what this means on the regulatory front, you talked a little bit about, what regulators are thinking about, but Do you feel interactions with regulators on manufacturing of gene therapies have changed from the past? And if so, in what ways? And then, and that applies both for US regulators and as well as those in ex-US geographies. And maybe I'll start with Fred and then go to Pierre and then Zandy.
I think this is a really important question for us. And I think in our experience at Taysha is, our opinion is that the conversation with regulators is always evolving. It's constantly evolving. If you have kind of a snapshot of what regulators' positions were 5 years ago, 2 years ago, 6 months ago, it's continuously evolving because they're seeing multiple programs from us. So I think from the field and learning from each of them as they review dossiers. And so I think what we've found is putting our information and our plans in front of regulators is really important in the form of, first pre-IND meeting, type C meetings, end of phase 2 meetings. I think it's really important, and just sharing our intentions. And I think the expectations seem to evolve. And I think we've been talking to multiple agencies. We've had 9 agency interactions for 5 programs for 4 agencies in the last 18 months. And we've learned immense amount of information from all of them. And there are, one certain agency might be focused in the CMC space on one particular issue. And, you have to have those conversations about their concerns. Another agency might be more focused on another issue. And so that's been a little bit of, it's been interesting, but it's also been a challenge because trying to meet everyone's expectations can become difficult. But I think it's been really informative and we've had really productive interactions. That's my thought. Some of our new guidance documents are helpful, but it's not quite specific enough. So I think going to the agency with your program is really, I think, important to have the correct expectation for, where we will meet with regulators when it's time for IND, BLA, or otherwise. So that's my thought.
Great. Pierre?
Yeah, great. I mean, those interactions with the regulators are really key. This is also good internally to align, the manufacturing with the research, with development, with regulatory, with quality before you go to the regulators. I mean, frankly, the know-how in the health authority has increased tremendously in the last few years. So they learn very quickly. Indeed, they learn from all our programs that we discuss with them. So I find the interaction always at very high level. This is not always completely clear, frankly, when coming back, okay, what needs to be done, right? The typical answer is, okay, you need to show, you need to have data, right? We knew that before going to the meeting. But this is usually the preparation, and the meeting itself is usually scientifically also always very interesting. But indeed, having the right regulatory affair people who can ask the right question and have the right interaction is really one of the key success factors in our industry.
Yeah, I mean, we're often asked the question, is the FDA just much stricter now than they previously were? And I don't think that is happening. I think what may be perceived as stricter is more and more companies becoming later stage in development, at which point there are higher and higher bars and hurdles that you have to get over in order to have pivotal material, file a BLA and get approved. So I don't think that there's been a change in the agency's requirements for, safe, consistent material. And I think agencies globally, as well as companies, are, as everyone has just said, with all of these filings, are learning a lot more about where we are currently and what's required. I will say that we have a pivotal program with our partner Janssen, and one of the things that has been most the greatest learning, there's been many learnings, but in that particular collaboration, is when you work with a company like Janssen, the focus isn't on making clinical material. It is on reliably producing material for a BLA for commercialization. And so along with the team at Janssen, we've now had regulatory interactions with, I think, around 15 different agencies around the world. We hear similar and different questions from all of them. We have teams answering those questions on CMC and QC. And these interactions have been going on since we were in a phase 1/2. So it's really iterative and it's really focused on making sure that your material is reliably made, that what you say your material is that material, and that has shown to be safe and effective in people. That's really what the agencies all over the world are looking for. And I don't think 5 years ago or today, you can really cut corners on that. And I think we're learning a lot more about how to demonstrate that to global agencies to make sure that they have comfort that that's what we're doing when we have products that we want to commercialize.
Great. So changing gears a little bit, as gene therapy programs have advanced through clinical studies and more programs have reached the late stage trials and are approaching the market, what have you noticed regarding manufacturing trends? For example, focus on local delivery or incorporation of automation or any other elements that you've noticed? And let's start with Pierre and then go to Zandy and then Fred.
Yes, as I mentioned earlier, at some point it becomes less about science, but more about technology and robustness. And so indeed, you mentioned automation. Once you don't need to do 2 runs per year for one product, but you need to do 2 runs per week, let's say, or 1 run per week, then you need to work in a different way. So you need indeed to add automation. You need to have robust process. You need to maybe use the same term we use in the more mature world of pharma or biotech. So, right first time, lean. You need to think about throughputs and not only about yield. At some point, it does not help to have a yield which is 10% higher if you cannot produce 2 batches in a row, right? So this is about making sure that you can industrialize your process. So such as not only a PhD can do the pre-culture, but that you can have almost cheesemakers that can do your process. I love cheesemakers. I'm Swiss, so cheesemaker for me are great. But these are the people that are great operators and know about hygiene and are able to, they're happy to work in biotech or in pharma. And these are the type of people that you need to have. So certainly automation, industrialization, the world of lean, visual management, how you make a big machine out of your manufacturing area. This is certainly one. The other one, maybe to finish on that, is data. I mean, we all speak about data. At some point you generate so much data in process development, but in manufacturing as well, every batch is a new set of data, and you need to be able to link the batch number of your media, your salt, with the deviation that you have, or process development data, and all this is a huge amount of data. And the question then is, how do you make those data readable? How do you generate information, and how do you make the right decision based on data? So that's already a struggle during the process development, but you have usually experts and you have maybe one program at a time. Once you start to have a huge amount of data because of manufacturing, this is important that you structure your database to collect those data in the right way.
I'm not sure what the trends in general in the industry are. I think that we worked very early on with regulatory agencies in Europe to build a facility and a process, which is what they wanted to see in the future. So that one of those things is single use and minimizing operator input, which is more or less what you were talking about with respect to industrialization. And we've incorporated that into our process and our facility, such that in our facilities, each of our suites is more or less like an independent little facility, its own air handling. The bioreactors come in and out when a batch is done. The same process is used. You can change scale over a 3-day period. The room is shut down, it opens up, you put new bioreactors in. So you have scalability and flexibility built into this system, which, because it's single use, allows for that without cross-contamination, as well as all the other design features of your facility. And really importantly, as one starts being in a pivotal program and starts looking forward to potential commercial supply, you know, not only the supply chain for the upstream and the analytics, but you don't want this huge, great market to be supplied by one site. So how do you then have 2 sites covered by the same QA? You know, one of the largest teams at Meira is QA and QC. QA and QC have around 60 people, and really preparing every aspect of the supply chain and your own supply to de-risk a potential commercial product. Because the last thing you want to do is to have a great phase 3, a drug approved, and suddenly you can't manufacture it. So all of the things that go into manufacturing anything apply to gene therapy as well. It's not just a research program, and you have to think about every single one of those, from your facility design, how your process fits into that facility with the minimum amount of potential risk points for making a mistake, how the documentation is perfect for every single batch, and then how do you make sure that if anything does go awry, you definitely have a second source supply, which is equally well controlled. So there's a lot to think about as you get into a pivotal, because that's really, you know, your commercial situation.
Great. And Fred, you comment
sure.
your end.
Yeah, again, just thinking about trends in the space, not specifically at one company, is, you know, I think we've all seen over the last few years, you know, as we have more programs moving into commercial stage manufacturing, you know, those learnings about, you know, scalable processes, those moving back. I think the fact that we have, you know, panelists that now are working in baculovirus, that was not the case 10 years ago. I think investing in those platforms that can be commercialized, and then it really across the board, scaling up processes. You know, now it's common to hear about production batches done at 500, 1,000 liters, 2,000 liters. I think looking at cell line engineering that would support that. And then kind of bringing in, like, I think that the way Zandy mentioned, bringing in some of your critical elements, your supply chain, plasmid manufacturing, you know, companies making that decision to invest internally rather than just buying this on the market because it's such a great risk. I think that's a real trend in the field. And then it's then really owning your analytics. I think that came up a couple of times already today. I think there are a number of CROs that provide testing services, but we've all found the, you know, significant delays as everyone is stuck in a queue, an assay goes down and then you're stuck and you're unable to release your batch. You know, really thinking about this end to end from design perspective, like what platform do I choose? How do I scale it? How do I support it from a supply chain perspective? And, you know, I also should mention, you know, some of the early programs in gene therapy were done in adherent culture. I think most companies have moved away from adherent culture and serum. I mean, I think there are so few of those instances that are in the field today. So that's one, I think I should have mentioned that up front. I think that's a big change for all of us. And I think that those are the important trends I've seen. And then more investment in more analytics. So I think we have really advanced tools that are coming online to measure things like empty to full, not just looking at the particle, but what's in it, how we look at it. Next generation sequencing, I'll give that a plug. I think there's some great work that's been done. And these are the questions the regulators are asking. What exactly is in your particle? Not, you know, you can't simply confirm that your transgene cassette is there, but is it full length? You know, what else is inside the particle? You know, AAV are really odd virological beasts that are promiscuous in the way they package things. So I think we need to answer all those questions. So that's the themes that at least I've seen in the conversations I've had around the field.
Fantastic. So I see we're just at about the 45-minute mark. So I think that's a great note to end this on. And I'd like to thank you all again for participating and for the fantastic discussion.
Thank you, Geulah.
It was a pleasure. Have a great day.